Hello, biology students, and we are here today to talk about macroevolution because we talked about microevolution last time, and today it is macroevolution. So, with speciation and macroevolution, this is one that gets a little more complicated with controversies, so we're just going to dive right in. So how new species evolve, mode of speciation, and principles of macroevolution. So first of all, macroevolution is evolution on a larger scale and best observed within the fossil record, involves the origin of species, also called speciation, and splitting of one species into two or more species, and final results of changes in the gene pools, allelic, and genotype frequencies. So that's what speciation really is. So with microevolution, we were looking at just changes within a population, just not really creating a new species, just like, a golden doodle, which is a poodle and a golden retriever. I mean, they're still a dog. They're not really anything different. Just creating a different dog breed, but it's still a dog. This is creating something completely new. One things we're going to see is that this takes lots and lots of time, and speciation is a creation of something entirely new. And one of the reasons we are not seeing this happen very often anymore is just that we have not been paying attention long enough to really observe this. We have only within the last 100 or so years put a name to this, and we have not been observing this long enough to actually pay attention to see it happen. So Linnaeus is often considered the father of taxonomy. He once hypothesized that birds and bats should be together in the same group because both have wings and fly. Now we know that birds and bats are different, and that birds are actually in their own group and bats are in the mammal group. And they are separated on the tree of life and in two separate groups based on other characteristics. Characteristics that we now are able to see because we have looked closer, and that birds are aves and or bats are with mammals. Every species has its own kind of evolutionary history, and we can classify that, and we've talked about classification before. All of the diversity of life on Earth has originated from the evolution of a new species. And if we trace things back far enough, everything has come from one original species. So species concepts refers to the different ways in which a species is defined. Based on analysis of diagnostic traits, distinguish one species from another. So there are different things that we can look at to help figure out, okay, what puts this animal in this group and what puts this animal in this group. Species can be distinguished anatomically by one or more distinct physical characteristics. Now, keeping in mind that it's not just animals, plants can be grouped into groups as well. So we're not just looking at animals here. This can be plants also. This method was used by Linnaeus, and most species are described this way. This is held up for 200 years. But bacteria and other microorganisms do not have many measurable traits. We can just look at them, but it is very hard to put some of these smaller things, especially, into groups. So some of these larger ones, it's a little easier because we can look at them and say, okay, well, yeah, this one looks pretty close. Going back to that scientific name, that helps to give us a hint. These all are called Rana in their scientific name. The scientific name consists of a genus and a species. So these all have a genus and a species. And the genus is all the same on these. So that means these are all pretty similarly related. So this is Rana burlanderi, and this is Rana sphenocephala, and Rana pippins. So Rana means they're all in the same genus, and since they have a different species name, it does mean they're all different species. But you can see they all have very similar appearances. They're all some kind of leopard frog, and they have calls that are different from each other, and they have different locations. So because of that, they are different enough. Also, they would not be able to interbreed, which is a key factor when dealing with different species, is that they cannot interbreed and produce fertile offspring. So that's a key factor with different species. So the evolutionary species concept distinguished species from one another based on morphological or structural traits. It was used to explain speciation in the fossil record and implies that members of a species share a distinct evolutionary pathway. They share common ancestors that had died off previously, and that those common ancestors had a trait that developed, and then those traits developed into something else, and so on over time. Since fossils don't provide information about color, soft tissue, or anatomy or behavioral traits, they are of limited use. Oftentimes we can find a fossil, but we have to kind of fill in the gaps about the rest of it. We don't know what color they were. We don't know what their tissue looked like. We really just know what their bones and their main structure looked like. We don't have a lot of that other fill in the gap kind of stuff. So here's some examples of what the fossils look like. I mean, we really just have the fossils to go by. We could guess what they looked like. We can kind of put it together and figure out the fill in the blanks based on their body structure. But we don't have a firm grasp on the rest. Did it have a dorsal fin? Did it have a flipper? Did it have a tail? What did its tail look like? Because we don't have a lot of those structures. Sometimes, if depending on the way it's laying, we can get some of those clues. The phylogenetic species concept is used to identify species based on a common ancestor. It is based on a single ancestor for two or more different groups. For you and your cousins, your grandmother is a common ancestor. So that kind of gives it like a perspective that is easier for you to grasp. You have a common ancestor just like animals and plants do as well. Populations of the same species breed only amongst themselves. So that is true because species can only reproduce and make fertile offspring if they are of the same species. They experience reproductive isolation from other such populations. Very few species are actually tested for reproductive isolation. A group of birds, collectively called flycatchers, all look similar but do not reproduce with one another, so they are different species. Leopard frogs live in different habitats, have a different courtship song, and are different species. So reproductive isolating mechanisms inhibit gene flow between species. They could live in different places. They could have different reproductive seasons. Maybe one reproduces in spring and one reproduces in fall. Maybe the organs themselves are not compatible. There are lots of things that could reproductively isolate them. Parazygotic isolating mechanisms and postzygotic isolating mechanisms. Prezygotic and postzygotic. So here we have pre-mating, mating, and fertilization, and behavioral, temporal isolating, and gamete isolating, and hybrid sterility. So prezygotic isolating mechanisms prevent mating attempts or make it unlikely that fertilization will be successful. Habitat isolation, species occupy different habitats. Temporal isolation, each reproduces at a different time, it's a different time of year. Behavioral, courtship patterns for recognizing mates differ, like frog calls being different, or a bird dancing being different. Mechanical isolation, incompatible animal genitalia or plant floral structures. Gamete isolation, gametes that meet do not fuse to become a gamete, like the egg and the sperm just don't match. So here are breeding seasons for a wood frog, leopard frog, and pickerel frog, and then the green frog and the bullfrog, they all mate at different times, so temporal isolation. Prezygotic isolating mechanisms for the blue-footed booby. They are not able to mate because of the different breeding seasons. Post-zygotic isolating mechanisms prevent hybrid offspring from developing or breeding. Hybrid inviability. Hybrid zygote is not viable and may die. So when the egg and the sperm hatch, this is not a viable egg and sperm. The offspring just cannot physically grow. Hybrid sterility, the zygote, which is the developing egg and sperm, may not develop into a sterile adult. Or may develop into a sterile adult. So this is a great example of a horse and a donkey. A horse and a donkey create a mule, and the mule is sterile. The mule is not able to produce. The only way you get a mule is by creating another mule with a horse and donkey. Again, great example. Mules are sterile. They can't produce other mules by mating a male mule and a female mule. So horse and donkey, make a mule. Only way you can get a mule is a horse and a donkey. So speciation is the splitting of one species into two, the transformation of one species into a new species over time. There's two modes: allopatric speciation. Microevolutionary process such as genetic drift and natural selection alter the gene pool of each population independently. When differences become large enough, reproductive isolation may occur and new species are formed. Geographic isolated populations of one or more species become different species over time. So this is like if there is a flood and suddenly a river divides two populations. It used to be one, and then the river separates them, and now there's two. Can be due to different selection pressures in differing environments. So here we see salamanders that got divided, and now they are in separate areas and they can't reach each other. And because they can't reach each other, this one looks like a mountain range, Central Valley barrier, a valley. Now they can't reach each other, and so they have developed into different species. So here we have some salmon, and the salmon have separated into different kinds of salmon, and they are sockeye salmon, and they are unable to breed. They've become different species. So sympatric speciation. One population develops into two or more reproductively isolated groups. There is no prior geographic isolation. Example is Midas and arrow cichlid fish. The arrow cichlid fish evolved from a population of Midas cichlid fish that adapted to living and feeding in an open water habitat. In plants, sympatric speciation often involves polyploidy, a chromosome number beyond the diploid number. Results in self-fertilizing species that are reproductively isolated from either parent. A polyploid plant can reproduce with itself, but cannot reproduce with the diploid population because not all the chromosomes would be able to pair during meiosis. So there are two types of polyploidy are known. Poly meaning many. Autoploidy occurs when a diploid plant produces diploid gametes due to non-disjunction during meiosis. Non-disjunction means not being able to separate. If a diploid gamete fuses with a haploid gamete, a triploid plant results. A triploid or 3M plant is sterile, can't reproduce, and cannot reproduce offspring because the chromosomes cannot pair during meiosis, so the chromosomes can't match up. Alloploidy is a more complicated process than autoploidy. Requires two different but related species of plants, hybridization followed by doubling of the chromosomes. So that all sounds a little confusing, but here's kind of a picture. We've got our diploid and our diploid. This one's got 14, this one's got 10, so they're a little bit different. They form a hybrid. The hybrid now has 24. So this one's not going to be able to do much because it's alloploidy. It's got 24. So it's a little bit different. It's still diploid. but it's not going to be able to do much because it came from two separate species. This one's, I mean, the species are kind of similar because they share a genus, but it created a different species that has 24. So adaptive radiation occurs when a single ancestral species rapidly gives rise to a variety of new species as each adapts to a specific environment. So you have one, and then that one gives rise to several. Several because, and it happens kind of quickly. Many instances of adaptive radiation involve sympatric species following the removal of a competitor, predator, or a change in the environment. So something happened in an area that causes like a restriction was removed. Something that was restricting populations from taking over, suddenly something that was holding it back before, now they can suddenly flourish. And maybe a new habitat was opened up. Or maybe a predator was removed that used to be there before, and now it's suddenly no longer restricted. So they are able to take over a new area, or where a predator was before, now it's not anymore. So it's opened up a new area for them. Allopatric speciation can also cause a population to undergo adaptive radiation. So here you can kind of see adaptive radiation in Hawaiian honeycreepers. So in this case, there are quite a few variations of honeycreepers that have suddenly taken off and expanded very quickly. And we've got all these different varieties of honeycreepers that have advanced in Hawaii. So convergent evolution. With this one, they start off as occurs when a similar biological trait evolves in two unrelated species. So two different things start off as two different things, but they converge to similar. And that's because they are experiencing like similar pressures and similar environments, but it could happen under two different environments. So even like two different continents. And they happen two different places, but they kind of become similar over time. Traits evolving this manner are termed analogous, so similar functions but different origins. Bird wing versus bat wing. They are both for flying, but they both take advantage of the air, but they happened under different kind of conditions. So this is opposite, or homologous is the opposite of analogous. So analogous traits are similar function but different origin. So similar function is they start off, or let's back up. Analogous means similar function but different origin. Bird wing, bat wing, insect wing. Homologous traits are similar but they evolved from a common ancestor. Wings of a butterfly and moth, since both evolved from Lepidoptera. Okay, homologous means traits are similar because they came from a common ancestor. Homo means the same. Okay, so here's convergent evolution. Convergent means two different animals got similar traits. Even though they were in different places, they converged similarly because they experienced similar conditions. Convergent evolution, similar traits even though they were in different places. So these two fish look similar even though they came from different conditions because they experienced kind of the same places. So Darwin's finches are an example of how many species originate from a common ancestor. So there was originally like one finch that came to the Galapagos Islands. But that one finch had offspring, or maybe there's a couple finches that were all the same. And they kind of spread out, and one of them ate insects, and one of them ate fruits, and one of them ate nuts, and their beaks all adapted to those different food sources. Over time, each species of finch adapted to a unique way of life. Beak shape and size are related to their diets. Increase or decrease in gene activity fine-tuned their beak morphology. So the cactus finch has a low level of this specific beak type and a high level of this specific gene to create a unique beak pattern that allowed it to eat this type of food. So the beak changed to depend on what the bird was eating. So just some showing the different variations. The beak changed for whatever the bird was eating. So insects or fruit or nuts or seeds or nectar, whatever, the beak changed to fit what the bird was eating. Just showing more of that depending on the two different types of genes and how it was expressed. Some evolution supports a gradualistic model. Evolution at the species level occurs gradually. Speciation occurs after populations become isolated. Each group continues its own evolution pathway, and the gradualistic model suggests that it is difficult to indicate when speciation occurred. It occurs gradually over very long periods of time. Some paleontologists support the punctuated equilibrium model. So again, all this happened so long ago, we're really not sure when or how. We've just got fossils to base it off of, and we've got our best guesses, but scientists are fairly sure that this is what happened. So the punctuated equilibrium model suggests that periods of equilibrium are punctuated by speciation. So we go through a time when things are stable, and then every now and then it's like, boom, evolution. Species can appear quite suddenly, is what this model suggests. Assembly of species in the fossil record can be explained by periods of equilibrium interrupted by abrupt speciation. So sometimes species change very quick, and then it levels off. And then it goes really quick, and then it levels off. Stabilizing selection can keep species in equilibrium for long periods. So again, kind of levels off, and then boom, big change. And then levels off, and then big change. So we have some gradual models, and then we have the punctuated equilibrium model. Genes can bring about radical changes in body and shape. Gene expression can influence development. Using modern technology, researchers discovered genes whose differential expression can bring about changes in body shape and organ. So this gene is in eye formation, this one is in limb, this one is repeated structure in the vertebrae. Research suggests these genes date back to a common ancestor that lived more than 600 million years ago. So they can find some genes that are similar in us as in other animals that indicate a common ancestor that was found in us and in other animals that, again, shows evidence of a common ancestor. Walter Gehrig and colleagues showed that mutations in the PAX6 gene led to failed eye development. So here are some other ones. The TBX5 encodes a protein transcription factor, turns on genes needed to make a limb during development. So if that one doesn't get turned on, you won't get your limbs. The HOX gene controls the number and appearance of repeated structures along the main body axis of vertebrae. And alterations in expression of the PITX1 gene causes reduced development of the pelvic fin buds and stickleback fish. So this is the PAX6 gene and eye development in all kinds of different creatures. One looks like a fly, obviously human, maybe a squid on that one, or fish. More studies of the PAX6 gene. So human evolution. The human DNA base sequence is very similar to that of a chimpanzee. There's only a few differences between the human and the chimpanzee. Human genome has about 19,000 genes. Predicts that differential gene expression, new functions for old genes, or both will explain how humans evolved. Natural selection acts on this variation. The survival of advantaged traits is favored in a particular environment at a particular time. Macroevolution is not really goal-oriented. There's not really a set goal that it's trying to achieve. It's just really trying to help us survive one day, year at a time. First probable member of the horse family lived about 57 million years ago. Horse evolution has been studied since the 1870s, and three trends were particularly evident during the evolution of the horse: increase its overall size, toe reduction, and change in tooth size and shape. So toe reduction probably helped with running speed, and change in tooth size and shape probably helped with its ability to eat. As more fossils were discovered, it led to recognition that lineage of the horse is complicated by the presence of many ancestors. We don't really know the direct ancestor of the horse, but we hope to find it and build on it, and that's why discovering more fossils is always important. Speciation, diversification, and extinction are common occurrences in the fossil record. So here we can see kind of the progression of the horse, so kind of almost small and dog-like. But as it progressed, we were able to see more of how the horse looked and how it progressed over time. This stands for a million years ago. Alright, well that's it for this chapter, and then we will see you for the next one. If you have questions, as always, you guys know how to get a hold of me. Bye, guys.